Welcome back to another video, the final one in this module, where we've been discussing various genetic mechanisms that regulate the expression of antigen receptors. And in the prior few videos, we discussed a process called VDJ recombination, which is a mechanism by which we determine the antigen specificity of a given antigen receptor, and in particular, the variable region of those antigen receptors, either immunoglobulins or TCRs. In this final video in this module, we are going to sort of look at a different portion of the antigen receptor within immunoglobulin, specifically that being the heavy chain, the constant region of the heavy chain, which in immunoglobulins actually confers effector function rather than antigen specificity. So class switch recombination is its own distinct genetic process which allows us to shape the way that immunoglobulins actually influence an immune response in ways that are targeted to the specific type of infection or the particular anatomical location in the body that immunoglobulin happens to be active in. So if we jump into looking at class switch recombination, I'll remind you that all B cells start their lives co-expressing two isotypes of immunoglobulin. Those were IgM and IgD. And you can see here that these are the B cell receptor forms, but there are also secreted antibody forms of these immunoglobulins as well, at least in the case of IgM. But during the naive state, IgM and IgD are co-expressed and either can serve as a B cell receptor. But B cells do not remain naive forever, and when they do see their antigen and become activated, they can undergo what we call class switching, in which they cease to express IgM and IgD, and instead they take on expression of one of the other available isotypes like IgG, IgA, or IgE. And I'll note that these other isotypes are expressed in isolation. They are not co-expressed with other isotypes. So the activated B cell that has seen its antigen, if it is in the right microenvironment, it relies on microenvironment cues, which we'll talk about in order to sort of decide which class to switch into. But assuming it sees its antigen and it gets the right sort of environmental trigger, it will undergo class switch to begin to exclusively express one of these other three, IgG, IgA, or IgE. And so we can begin to look at how that occurs mechanistically, especially at the genetic level, by looking at the way that the constant heavy segments of immunoglobulins are encoded, or where we find them within the heavy chain locus. So the heavy chain locus looks something like this. We've been looking more upstream in our past few videos at the V, D, and J segments that we use, again, to put together the variable portions of the heavy chain or of the immunoglobulin in general. But remember, we're in the heavy chain locus right now. But all of these additional segments, which make up the C-terminal end of the immunoglobulin, which are here colored with these different colors in this figure, they are going to be encoded by other segments, which are not recombined in the sense that VDJ segments are recombined. They rely on conserved exons that are encoded downstream, and that can be spliced in various ways to determine, or in order to be added to the previously recombined VDJ. So the VDJ is encoding the variable segment of the heavy chain, whereas these exons, or at least these genetic regions that you see, these loci downstream, are what are going to encode the constant heavy chain. And what I want to point out to you, I said that IgM and IgD are co-expressed by naive B cells, and the reason that they are co-expressed and the other isotypes are not yet expressed is because the exons that encode the constant segments associated with IgM, we call that region constant mu or C mu, and those exons that encode the constant segments of IgD, which we refer to as constant delta, they are immediately downstream of the J segments in the heavy chain locus. That means that they tend to be, or that they're close enough to be included in the primary transcript as transcription proceeds through this region. And so they can be spliced into the final mRNA molecule. In contrast, these other sets of exons, which you see here associated with the other isotypes, they are a bit too far away, at least for right now, to be successfully transcribed and therefore to be successfully spliced into the final molecule. So the distance of within this chromosomal DNA sort of prevents the expression of other isotypes beyond IgM, which again is associated with constant mu, and you can remember that mu starts with an M, or at least it has the sound of an M, so that's where IgM comes from, is its association with mu. Similarly, IgD is associated with delta, the Greek letter delta. For these other isotypes, what you can see is actually in humans, there are even subclasses of these isotypes. So for IgG, most humans are able to express 4 subtypes of IgG, IgG1, IgG2, 3, and 4, and they each have their own set of constant segments, which are found together on the chromosome. So you can see here you have a C gamma 3 region, which will, if it is expressed, will give us an IgG3 subtype immunoglobulin. Similarly, there are 2 subtypes of alpha constant segments, and so this would encode IgA1 and IgA2, while humans have a single functional constant epsilon, which will encode the IgE variant. You can notice that humans also have a pseudogene for constant epsilon, which is not functional. And therefore is sort of kind of functionally irrelevant. We don't have multiple subclasses of IgE, but we sort of have the leftovers of what was probably formerly a functional additional constant epsilon, meaning a former epsilon isotype that maybe we used to express in the past, but has now been rendered a pseudogene. So for the purposes of the lecture today, I'm not really going to focus too much on the subtypes of gamma and alpha. I really just sort of want you to just think about the fact that we have 5 isotypes, and these 5 isotypes are encoded by discrete constant heavy segments in the heavy chain locus, and we can just sort of imagine that there's a single gamma and a single alpha just for simplicity's sake as we go over the concepts today. But I at least want to sort of inform you that there are subtypes, each of which have their own distinct set of exons, as you can see here. But let's start at the beginning. As I said, all naive B cells begin by co-expressing IgM and IgD, and that is because they have access initially to the constant mu segments and the constant delta segments, which are the closest. They're the most proximal segments to the J segments in the heavy chain locus. But we co-express them. So how does that co-expression work? Well, that is simply a process of alternative splicing. So we saw before that immunoglobulin molecules can be expressed using the same genes that can be expressed in 2 forms, as either a B cell receptor or as a secreted immunoglobulin. And we saw that that was determined at the transcript level by splicing in of either a hydrophilic final segment, which targeted the molecule for secretion as an antibody, or a more hydrophobic segment, which targeted it to the membrane to serve as a BCR. A very, very similar concept of alternative splicing determines whether any individual transcript, primary transcript of the immunoglobulin molecule will be IgM or be IgD. And so if from that primary transcript, through the process of splicing, we keep the exons associated with constant mu, they are incorporated into the final molecule, which gives us an IgM immunoglobulin. Or we might instead splice in the exons associated with constant delta. And in that case, we would exclude the exons associated with constant mu, and that would result in an IgD molecule. And this is essentially a random stochastic process that happens for each individual transcript. And so some transcripts become IgM, some transcripts become IgD, and therefore they are co-expressed. And a naive B cell will have both until it undergoes class switching. But the expression of the other isotypes is not accomplished via alternative splicing. They are not included in the primary transcript because, as I said, they're a bit too far away for that to effectively occur. So there's a different way that we sort of gain access to those exons in order to include them in the immunoglobulin mRNA, and that will be what we discover. It will be called class switch recombination. So recombination should tell you that's a different process than mRNA splicing. Okay, so having explained that, let's get into what actually occurs in order to access those other isotypes. So a point that I want to make is isotype switching, class switching, is not a random process. It's a very directed process because class switch is usually done in a way that facilitates a targeted immune response against a particular type of pathogen. And what I want to point out here is that there's sort of three major types of responses. As usual, this is a bit of an oversimplification, but I think it's at least a good place to start for understanding these concepts. In general, the types of immune responses that we mount to viruses and bacteria are sort of classified with the umbrella term as a type 1 immune response. This includes a variety of innate and adaptive immune processes or responses that are specialized or are especially good at fighting viral and bacterial infections. One of the key features of a type 1 immune response is high expression of the cytokine interferon gamma. And interferon gamma does many things as part of a type 1 immune response, but one of the key features, one of the key things that it does is it influences or it promotes class switching into IgG. And as we'll see, IgG has effector functions that IgM does not have, and those effector functions associated with IgG are especially good at promoting antiviral and antibacterial effector responses in other cells and in the tissue more broadly. So it makes perfect sense that the cytokines that we tend to produce during these types of infections, those cytokines will also serve as signals that cause us to make antibodies, which are also good at fighting the same type of infection. And so these things are all functionally linked in ways that have been guided by evolution. Similarly, during a parasitic infection, the types of immune responses that we use to fight parasitic infections are distinct from those that fight bacterial and viral infections. And in response to that, typically different cytokines are expressed, including a very important type 2 cytokine known as IL-4. And IL-4, for example, tends to induce class switching to IgE. And as we will see, IgE also has particular effector functions. It activates particular Fc receptors, especially that unlock anti-parasitic effector mechanisms. And so there's also a targeted response that takes place there. Finally, B cells that find themselves at mucosal surfaces will encounter cytokines enriched at the mucosa. This includes, among others, but probably most famously, IL-21 and TGF-beta. And the high concentrations of these two cytokines at mucosal surfaces influences class switching into IgA. And IgA is particularly enriched in the mucosa, and it has some special features which allow it to survive in the harsh environment of the mucosa and interact especially with the bacteria that tend to colonize those areas, especially opportunistic bacteria that would like to invade the body through those surfaces. And so the direction of class switching towards IgA at mucosal surfaces also serves a very important immunological function. So the big idea here is that inflammatory microenvironment is going to be an important signal that is going to shape B cell class switching. But assuming the B cell is activated, it sees its antigen, and it receives one of these cytokines, let's look at what actually occurs in order for class switching to be accomplished. So here we can start with our naive B cell, and if we zoom in a little bit further on the heavy chain locus, we previously saw that there were these genetic regions that we called constant mu, constant delta, and so on. But what I'm adding to the figure now are these additional genetic regions, which we refer to as switch regions, which are denoted as S mu, S gamma, S epsilon, and so on. Notice that there is no switch delta, however, and that constant mu and constant delta are still found right next to each other. And this is another reason why they tend to be transcribed together and therefore co-expressed. But for right now, what I'll point out is that in a naive B cell, the only switch region that is active, or the only switch region through which transcription is occurring, is the switch mu region. And that is because this is the only area that is transcriptionally active because the cell is only expressing mu and delta, which are immediately downstream. However, in order to accomplish class switching, a major thing that needs to happen is that we need to induce transcription through one of the other switch regions. And so for this example, I'm going to talk about the class switching into IgG, which will require the expression of constant gamma constant segments. And so remember that there are multiple subtypes. Here I'm just sort of pretending that there's a single constant gamma, just in order to illustrate the principles at work here. But just with that caveat, notice now that transcription is active both at the switch mu region as well as the switch gamma region. And and this is occurring because this B cell is not only seeing its antigen, but it finds itself in an environment rich in interferon gamma. Remember that interferon gamma, as part of a type 1 immune response, is going to influence the cell to class switch into IgG. And so one of the ways that it does that is by promoting transcription through the switch gamma region. And the reason that transcription is so important is because transcription necessarily opens up the double-stranded DNA so that RNA polymerase can bind among other molecules. And that opening up of the DNA also exposes the DNA to a unique enzyme that is only expressed in B cells called activation-induced cytidine deaminase, or AID. Again, this is uniquely expressed in B cells, which is one of the reasons that only B cells do class switch recombination. What we say is that when the DNA opens, because transcription is proceeding, that renders the switch regions that are being transcribed sort of susceptible to AID. It gives it AID access to the DNA as it's being opened. AID is actually a mutagenic enzyme, and it's going to induce mutations in the switch region, which ultimately are going to induce recombination. So let's take a look at how AID, the deaminase activity of AID, accomplishes class switch recombination. So if we zoom in yet even further now on these two regions, you can sort of imagine that we are now in both the switch mu region and the switch gamma region. Activation of the B cell has induced the expression of AID. The DNA in these switch regions has opened because transcription is occurring. And now that we also have AID expression, it is going to proceed to induce mutagenesis. And the type of mutagenesis that it induces is the deamination of cytidine bases into uracil bases. So what you can see here as you go from left to right, you start with normal DNA and notice that there are some Cs, there's some cytidines here. And now in this image, all of those cytidines have been converted to uracil. The consequence of that is that we now have mismatches. Uracil is associated with RNA typically. And so to have uracil in the DNA triggers the process of mismatch repair, because now all of these Gs are suddenly paired with Us, which they are not actually complementary to. So now that the cell's mismatch repair machinery is recognizing these mismatches, it's going to want to remove them. And there are two enzymes in particular, U and G, and as well as an enzyme called APE, which are part of the mismatch repair machinery. And simplifying a little bit, what I'll say is that it wants to, these molecules will excise those uracils. They will basically just eat them out, clip them out. And this induces a combination, several types of DNA damage, including double-stranded breaks, which hopefully you can appreciate in this cartoon here. There are breaks on both sides of the DNA. And those double-stranded breaks are what are going to be really important for inducing the downstream processes associated with recombination. So taking all of this together, we're inducing double-stranded breaks in both switch regions through which transcription is active, both in the switch mu region as well as the switch gamma region. And this is important because for switching to occur, we sort of need two switch regions to be damaged in this way, and for these double-stranded breaks to be induced in both. So again, right now we're imagining that we're in a situation where we want to class switch into IgG, and so we've induced transcription through the switch gamma region through exposure to interferon gamma, and this is what has occurred. So assuming that we induce these double-stranded breaks in both of these switch regions, What happens next is that the double-stranded break repair response sort of kicks in. And we won't cover all of the enzymes involved in the double-stranded break repair response. Many of them overlap with the enzymes involved in VDJ recombination, although not all. But suffice it to say that those double-stranded breaks in both of these switch regions, which I've now tried to indicate with these sort of dotted lines around the squares around the switch regions, the cell wants to repair those double-stranded breaks, and the way that it will do so is to bring the regions that have been broken close together and synapse them, as we've seen previously in other types of recombination. Actually bring those two pieces of DNA together. What's going to happen is that as it brings those two pieces together and synapses occurs, the intervening DNA, again, will be looped out and eventually cleaved off. And so as the double-stranded breaks are repaired, what occurs is that DNA between the breaks is removed and is eventually recycled, and now is no longer available to be expressed. We've removed it, again, from the germline, from the somatic DNA of the B cell. This is a permanent change to the DNA. The switch regions remain, but the switch regions themselves do not code. They are only used to sort of guide the cell in terms of where the process of synapsis and cleavage should occur. But notice now that even though the switch regions remain, the exons, the actual coding exons that are now most proximal, that are closest to the VDJ segments, are now the exons associated with constant gamma. So now that constant gamma is the closest to VDJ, it will be transcribed and included within the mRNA that will actually be translated to give us an immunoglobulin. So now this cell has successfully class switched into IgG. And a very similar process would play out if we wanted to class switch into IgE or IgA. Those cytokines would induce transcription through those switch regions, either switch epsilon or switch alpha, and all of the intervening DNA would be cut out between the two active switch regions. And if we were class switching into IgA, we would want to bring constant alpha closest to the VDJ through the same process. If we wanted to class switch into IgE, we would want to bring constant epsilon into the same position immediately downstream of the VDJ segments. And that is how class switch recombination occurs, again, in a nutshell. So we can close this lecture with one slide, which will just help us distinguish a little bit, I hope, between these two types of recombination that we've talked about in this module. The first several videos focused on VDJ recombination, and I want to point out that these two types of recombination, VDJ recombination and class switch recombination, are important for putting together or influencing the shape, the final shape of different regions of the immunoglobulin molecule. VDJ recombination is important for, and you can notice that VDJ here is in blue, it is important for determining the structure of these blue segments on the immunoglobulin molecule, and those are going to be the variable light chains, which might be either V kappa or V lambda, as well as the variable heavy chain, which remember are the sort of inner variable segments, and the light chains are the outer variable segments. The key enzyme that accomplishes VDJ recombination is RAG, recombination activating gene. And again, the functional impact of VDJ recombination is to confer antigen specificity on the immunoglobulin molecule to determine which antigen it will bind. And of course, that's very important. In contrast, though, class switch recombination determines the constant heavy region of the immunoglobulin, those subunits here shown in orange. And it, instead of using RAG to initiate recombination, it uses the mutagenic enzyme AID, which deaminates cytidine into uracil, which eventually produces double-stranded breaks, which trigger a different type of recombination process, which will determine which heavy chain isotype is incorporated into the final immunoglobulin molecule. And that is important because it will confer different effector functions on the immunoglobulin molecule. IgE, of course, activates Fc epsilon receptors, just as an example, and Fc epsilon receptors are good at inducing the degranulation of cells like eosinophils and mast cells, which again are very strong anti-parasitic effector cells. whereas a class switch into IgG unlocks effector mechanisms like classical complement cascade or phagocytosis. Those are more targeted to a type 1 immune response against viruses and bacteria. So these are examples of the way that class switching changes the effector function of an antibody in ways that make sense depending on what type of infection is occurring. Okay, we can summarize now. We saw in this video that immunoglobulin constant heavy chain segments are encoded by discrete gene segments, and that the exons associated with the constant heavy chain are downstream of the recombined VD and J segment. That's actually a point that I did not quite say, is that class switching is always happening long after VDJ recombination. VDJ recombination happens during B cell development, and B cells do not survive unless they successfully recombine a receptor using VDJ, whereas class switch recombination occurs later in the B cell's life after exposure to its antigen. And as we see, that class switch recombination does not influence antigen binding, but instead is important for conferring effector function. We also saw that naive B cells co-express IgM and IgD during homeostasis, and this is mediated by the constant mu and delta heavy segments that are immediately adjacent to the recombined VDJ, and that they are co-expressed because they are both available to be differentially spliced into the RNAs of the immunoglobulins in the naive B cell. But if a B cell becomes activated, it begins to express AID, and if we have the right cytokine in the environment, that is going to induce the process of class switch recombination, which is a permanent alteration of the DNA that is going to remove intervening constant heavy segments and confer expression of a new isotype. So that covers it for this module where we've talked about antigen receptors. I'll see you in the next module where we'll take a much deeper dive kind of into the other side of the equation, and we'll talk about MHC and antigen presentation. See you there.